Amazing. Okay, so first things first. Teaching everybody how this session is going to operate. On every slide, there is a who's coming up next. That person should come to the side to get ready to talk. The purpose of this is to give us all a chance to hear a short few-sentence pitch preview of what you'll be talking about at your poster so that we have a little taste of what we'll be looking forward to tomorrow and Thursday. So while Charlotte, who is here, comes up to the stage, let me just say, I put this together in just a few minutes earlier this morning from the list of posters I had at the time. If your poster wasn't included, please just come up at the very end. And I used large language models to generate this whole presentation. So we will see our AI overlord's interpretation of what your talk title is as we go. So I'm excited to find out.
Thank you. I'm excited. Oh, okay. Interesting. Okay. So hi, everyone. The subject of my poster is very different, as you would guess from this. But the subject of my poster is really self-folding and unfolding of thin multicellular sheets. And there is flocking involved, which is why you might see a flock up there. But so as you may know, folding and unfolding is very important in biology, particularly in multicellular biology and in the case of morphogenesis. And in my poster, I'll tell you about sort of a unique case of self-folding and unfolding exhibited by a very simple animal that's called Trichoplax adherens, which was mentioned this morning. And Trichoplax is extremely simple. It has no brain, muscles, or neurons, but it does have a dense ciliary carpet, which does exhibit flocking dynamics and is enables the animal to walk on rigid substrates. And so in my poster, we studied how folding and unfolding emerges from the collective dynamics of the ciliary carpet, and we discovered sort of an interesting coupling between the folding state of the animal and the flocking of this ciliary field. So if you're interested, please stop by.
Amazing. This can be a self-working system. Anyone who wants can push the right arrow.
No. Your language model failed.
Just a swatch of color.
Difficult. So I thought of showing some of the activity of my group. So we work at the interface between the biophysics of unicellular organisms and robotics, the idea of trying to replicate what we learned from biology into artificial systems, in particular control of shape and locomotion. So here I have a proxy. We've talked about bacterial flagella recently, but there's also eukaryotic flagella. They drive motion by bending, so sperm cells work like that. And then there's other forms of motility, not flagellar, but with the shape changes of the whole body, amoeboid motility. So imagine this doing something like that, okay? And so we work on this at the level of biological system. You will find in the poster two examples. One has to do with mucociliary transport, so the way waves can generate transport of a fluid and how these things can self-organize. And then the other thing is how organisms can manage to produce these complicated shape changes that, for example, entail the change. So geometrically, this is a cylinder, no Gaussian curvature, and the great feature of this is that it's a bulge, so it's something with non-zero Gaussian curvature, positive Gaussian curvature. How does this emerge? And what is the mechanics of systems like this? So we try to be quantitative, and so we like to ask questions like, imagine that I do this, what will be the height at which this will jump, okay? At the artificial level, we have a similar interpretation or rendition of this in two systems. One is, so they are both, so while the biological systems are powered by molecular motors, our artificial systems are powered by light. So they are liquid crystal elastomers powered by light, and we study self-oscillations produced by uniform lighting and if you light this, you can produce an oscillating system that does this. Or we've studied a medusoid, so something that swims like an ephira in real time. So if you're interested, please stop by.
And if you see me standing up, that means it's been...
Okay, well, that's not quite the image, but anyway. So the question I'm going to pose with my poster is to think about what are some general ways of designing metamaterials. I think there was a lot of examples presented today, and I'm going to approach this question by thinking about the symmetry properties of ordered metamaterials and thinking about how do they affect response properties. The response property I'll be looking at in the poster will be wave propagation, and I will be using sort of tools from group theory to talk about how does the propagation of elastic waves changes as you change the symmetry of these soft crystals. And it's interesting to also note that these soft crystals, it can change and cause buckling within these crystals by deformation, which also again causes change in symmetry, causing a change in material properties. So if you're interested in sort of thinking about how some of these methods work, please do come by the poster. And yeah, that's...
Okay, almost. So I'm going to present like kind of a tasting menu of some of the current ongoing projects in my lab on active biomimetic matter. We do experiments with purified proteins, hence maybe the petri dishes. And it's a tasting menu, so there's going to be a little bit of everything. If you like actin, we have some projects about actin treadmilling, and here we'll talk about anti-chemotaxis, flocking, and emergent beating. If you like microtubules, we do experiments with kinesin and microtubule bundles about active interfaces and like defects and dynamics in 3D active nematics. If you like DNA, we look at liquid-liquid phase separation, so like droplets of DNA that are like coarsening inside an active fluid. And finally, if you prefer like bacterial proteins, we looked at pattern formation in 3D in min-ED systems.
What? It actually gave the correct title? Yes. I am indeed going to talk about nonlinear and non-Hermitian mechanics of living non-reciprocal solids. So my work is basically going to be on an experimental system, which is the supersaurus system of living starfish embryos, crystals formed of living starfish embryos that were discovered in Nikta's lab by Zerhan, who's also in the audience. We've collaborated with Johan Dunkel's group to develop a data-driven method that allows us to characterize these materials. What we found is that, interestingly, these systems exist not in one, but in two stable states. The one is, the first is a fluctuating crystal, which, if you look at the dispersion relation that our method gives us, looks exactly like a bona fide linear odd elastic solid of the type that Vincenzo and Conata described earlier today. But the second state is an oscillatory state, which is actually quite different, and in fact, it shows the existence of optical bands in vibrational modes, in spite of the system being a 2D triangular lattice with only one atom per unit cell. I will not tell you why right now. Please come to my poster to know why. But we also can quantify nonlinear interactions between these optical and acoustic modes. And we are also very excited about the prospect of looking for non-Hermitian effects in this system, which our method in principle allows us to extract. And I know that there are many people in the audience who know much more about that stuff than I do. So this is very much ongoing work. Please come to my poster and give me some feedback, and be happy to have interactions. Thanks a lot.
Luis, going once. It's going twice. Intrinsic and extrinsic decision making. Very good. Par.
Okay, thank you. The image is not descriptive. Oh, that's not me, sorry. Yeah, still not. I know the theme is active solids, but I'm going to talk about phase separating fluid systems. This is in particular about phase separation between an active and a passive fluid. This is inspired by experiments with microtubule kinesin-based active fluid phase separating from just a passive Newtonian fluid. So I'll show you how the interfaces in these systems behave very differently from some passive thermally agitated interfaces. And you develop these curvature asymmetries. And moving away from the experiment slightly, we'll see how you get these system-spanning filamentary phases of the active fluid, which form these dynamical networks that sort of arrest and do not coarsen any further. And there'll be cool pictures about the 3D system, which we do not understand very well yet. So please come to my poster to see that.
Okay, hello. I worked on the isotropic nematic transition for semi-flexible polymers, which is a first-order transition. Depending on the flexibility of the polymers, they go from this isotropic state to this nematic state. And we want to look, if we introduce activity in the system, what happens to this transition. And what we found for low activity, that this transition changes to higher density. But if we increase the activity even further, we get this more dynamic state, where instead of a first-order transition, we get a more continuous growth of the nematic order. And we also looked at how the effect of the activity is on the conformational properties of polymers at different densities.
Hello. So in my poster, I'll be discussing some work that we've done on a model of a tissue monolayer. And so the formulation that we've used is this multi-phase field model. So you can think of each cell in the tissue as being a deformable active particle. And we've studied the solid to liquid transition as a function of the motility of the particles, as well as intercellular friction between the cells. And so we find that, interestingly, near the solid to liquid transition, there's the emergence of two kinds of order. One is local hexatic order in the positions of the cells, as well as local nematic order in the elongation axes of the cells. And both of these things have also been studied in experiments on epithelial cell monolayers. And finally, you can see how we find a correlation between defects in the nematic structure and the hexatic structure in our model system, and we propose a way in which this can relate to the mechanics of the tissue.
Hi. So the subject of our study is the emergence of metachronal waves. The subject has been well known that cilia with asymmetric strokes can generate the metachronal waves, which are plays important role in circulating fluids, for example. But recently, an experiment has shown that symmetrically beating nematodes called T-acetate can also generate the metachronal waves. So by using elasto-hydrodynamic models, we investigated the underlying mechanism of the generation of metachronal waves. Even if they are beating symmetrically, by using misalignment, they can effectively break the symmetry even even under the stronger strong dominant hydrodynamic interactions. So by tilting their bodies against the well, they effectively can break the synchronous motion and generate the metachronal waves. So by increasing the tilting angle, we found that at a certain finite tilting angle, the thermodynamic efficient is highest. So it is we found that it's quite interesting. Okay, thank you.
That's pretty close. So I'm being rewarded for having an overgeneralization for a title. So epithelial cells organize themselves in tissues, which are continuous cell monolayers that in which cells fill the whole space they're given. And this is important for their barrier function. On the other hand, it's very important that cells move in these tissues to perform some vital biological functions, like, for example, morphogenesis or wound healing. Which begs the question, how does one move without space? And cells have this ingenious way to do it, which is called cell intercalation. And here are my props. Imagine that my fingers are cells, and they want to move up and down, but there's no space. So instead they... Yes, please. Thank you. So what they do instead is they push inward. Thank you very much. They push inward, and they create this ladder intercalating and pushing each other up and down. So this is called cell intercalation. End of prop session. At the same time, mostly cells are hexagonal, and this order is usually disrupted by pentagons and heptagons, which do not allow it to extend to the rest of the system. So what we did is we used a hydrodynamic theory of active exarctic liquid crystals to show that the flip side of cell intercalation is the unbinding of dislocations in active exarctic liquid crystals, which are pairs of pentagons and heptagons. Now, do you find this interesting or perhaps preposterous? Please come to my poster and we can discuss all about it. Thank you.
All right. I don't know whether it's close or not, in my group, we work with these programmable self-propelled robots, which have various sensors like light sensor and infrared sensors. So they are pretty smart. They can sense obstacles and react to light signals. But in this poster, the question that I'm trying to solve is how do organisms attain motility, right? So you must have seen videos in the morning like starfish crawling on the surface of the ocean floor, or even millipedes. For me, the simplest organism is the Chlamydomonas, which has only 2 beating flagella, but it still moves. And it more like does a run and tumble motion, right? So motivated by that, we created our own compound robot, which are 2 robots connected with a rigid rod. And individually, both the robots are doing active Brownian motion. And we saw that within a given very fine parameter phase space, the compound robot starts behaving like a run and tumble. And if you are interested in knowing how that happens, please come visit my poster. We have simulation results as well, and we draw comparisons with Chlamydomonas system as well. And it's still very ongoing project, so there are still many puzzles, but you are welcome to visit my poster. Thank you.
All right, let's see the generate image. Oh, okay. So yeah, this maybe looks like embryo on the top view. So in this work with Nikta and your both in this conference, we try to address this question. In non-equilibrium, we know, as we saw earlier today, that we can have non-reciprocity. Now, how can this non-reciprocity reshape the collective behaviors of experimental active matter, such as the starfish embryo system, which without, let's say, explicit non-reciprocal interaction, already does very interesting thing that they self-assemble into a giant crystal, and they will do this collective rotation. Now, what we do is that instead of having single type, we have starfish embryo siblings, which exchange non-reciprocal interaction, such that the younger chases the older, while the older runs away from the younger. And now, if you collect 1,000 of such embryo siblings together, what they do is, first, instead of collective rotation, they do collective translation, as we saw in this talk just ago. And also, instead of forming one giant cluster, they form multiple small clusters, which is very dynamic. So to understand this, we used Bayesian inference to learn the interactions between these two embryos, and which was not only non-reciprocal, but also was able to recapitulate both of this collective polarization and let's say multiple clusterization. Okay. And I'll talk more about the posters if you come to my poster. Thank you.
Okay, that's an interesting interpretation. So the aim of this work was to generalize active particle models to curved surfaces. Diffusion and random motion on curved surfaces is a ubiquitous setting in biology, the most prominent example being probably membrane proteins. And life on curved surfaces is rich, beautiful, and sometimes quite complicated. So I guess all of you might know that if you move around on a curved surface in a loop, you generally don't go back to where you started. And this manifests in inertial dynamics by a coupling between rotational and translational motion, which we explored in this paper. But to go further, we would like to add noise to these systems as well, which is already quite intricate because you have to make sure that when you change your coordinates on your surface, then your noise also changes appropriately. And finally, what we wanted to do is to take the overdamp limit of these equations, and that's also usually quite subtle because it's a stochastic system, and in such stochastic systems, this limit can have some surprises. So we performed all these rigorously and in a mathematically consistent way. So if you want to know what these equations look like, then check out my poster at the session.
It cannot make sense of my title, which means my job is safe from AI for now. So hi, I'm Toshi. I'm broadly interested in proliferating active matter, and I'm going to be presenting a study this week which tries to understand how nematic order develops in a system that can grow, that can jam, and that interacts with an external field. We have an experimental realization of the system, which is a bunch of elongated fibroblasts that grow on top of a liquid crystal elastomer, the substrate being this external field that biases the order of the cells. So what we know both experimentally and from our theory now is that the final order, that means the order and density at jamming, strongly depend on where you started from, how much density there was when you plated the cells. And interestingly, the theory also shows that this order-disorder transition can be continuous or discontinuous as you change the magnitude of the field, depending on, again, what your initial condition was. We have some cute dynamical system stuff going on, like a line of fixed points, which changes its stability in between, and this change in stability gives you this first-order transition and sometimes the second order transition. And I don't understand all of it, so it would be great to talk to all of you so that we can develop a better understanding of this. Thank you.
Good evening, everyone. So this is Soni Prajapati. Actually, hello. Yeah. So basically, we are like doing a modeling. The work is kind of simulation work. So we are using the Brownian particles, and we are there is a background flow also. And what is the effect of that background flow on this active Brownian particle? That all we wanted to know, because in nature we see that this, like whatever biological particles are there, mostly they are surrounded by some fluid. So what are the effect due to fluid on this kind of particles? So that is what we are trying to explore it. Also, it also depends what is the cell propulsion velocity of these active Brownian particles as compared to the background. So whether this background flow will affect the dynamic or motion of this active Brownian particle or not, that also very interesting to know. Apart from that, what will happen if there is a comparable velocity of this active Brownian particle as well as a fluid? So this is all we are wanted to know, and we can learn more in my poster. So please do visit. Thank you very much.
Okay. All right. Hi, everyone. I'm Zeb Brockman. I am a professor at the Georgia Tech Physics Department, and I am going to be talking to you really quickly here about fundamental principles of flexible solids. And I would like to thank Dan and all the organizers here. I'm sure everyone would as well. Solids are important, and they're also very well understood. I know whatever this table is made out of, I know that when I lean on it, I'm going to have patterns of stress that develop, and when I give it a whack, I'll get sound waves. And something that's not as well understood is flexible solids. Flexible solids do all sorts of weird different things, even at relatively low energies. In fact, they're really hard to understand using the analytic techniques that physicists usually use. I can't predict exactly what's going to happen when I apply even relatively mild stresses to most flexible solids. And something that's different here is we have a community now that's gotten interested in changing the rules with autoelasticity, with anti-Hermitian interactions, with time delays, with all that. And the way we're changing the rules is we're talking about flexibility as being a specific type of deformation. Maybe it's bending, like for a thin sheet. Maybe it's dilation, what I'm going to be focusing on in my poster. But it turns out that when you have those specific rules, you can take a global mechanism, so a global dilation in this case, and you can say, what if I promote that to a mechanism field, something that dilates different degrees to different places, and that gives you a playground that can really give you lots of different transformations that are also very rational and predictable. So that's the general principle that I'll be talking about in the poster. Hope you stop by. It's taking up too much space, so we have to justify that. All right. Thanks.
Hi, everyone. So in my poster, I will be talking about my current research project, which is basically understanding shape change in contractile ectomycin gels. So in cell cytoskeleton, shape change happens due to activity of myosin motors, which bind to the actin filaments and produce contractile forces. So this project is motivated by in vitro experiments on contractile ectomycin gels made up of actin monomers, myosin, fascin crosslinkers. So these gels with same composition but different aspect ratio basically contract due to myosin activity and give rise to different 3D shapes like dome, saddle, wrinkles. So so we are trying to come up with a theoretical model that can explain those different 3D shapes. So in my theoretical model, I have like two main things, active stress and orientational order parameter, because in experiments, we also observe different amount of alignment of actin fibers at the gel periphery. So in theory, so in steady state, we can solve the minimal continuum theoretical model and explain the displacement and strain profiles as found in the experiments. So our theoretical model can explain a few features of the strain and displacement curves as obtained in the PIV. We are still trying to understand the dynamics of shape change, so currently we are trying to do the poroelastic simulations for that. If you are interested, feel free to stop by. Thank you.
Hello everyone, from Petya Vlochowska from Northwestern University. I'm in the Department of Engineering, Science and Applied Math, and this is just to say that active matter is everywhere. And I actually brought two posters from my group because it's like having two children and not being able to decide which one is your favorite. So one of them is going to talk about how, if you put an active particle in a container, how this active particle can drive the container to move. And the idea is being that now if you can program the trajectory of the active particle, you can also direct and have a directed motility of the container itself. So if you want to find out more, come, and you're going to see the beautiful experiments and theory coming from my group. And the other one is going to be about turbulent-like motion of motile colloids that are moving individually like bacteria. So these are the so-called Quincke random walkers that are kind of a new version of the Quincke rollers that were developed by Denis Bartolot's group. And what I'm going to show you is that experiments which show very intense bacterial turbulence-like flows. We have spectra that we don't know what we can learn out of them. And we also have some indication of structural memory, which I don't quite understand too, which the flow within one cycle gets completely decorrelated. However, the velocity fields between two subsequent cycles, when you are correlated. If you want to find out more about the structural memory, come talk to me, and thank you for your attention.
Hi everyone. My project is on a topological mechanism for length exploration in microtubules. So microtubules exhibit dynamic instability, which means that they can stochastically switch between phases of slow growth and rapid shrinkage. So this switching behavior is used for target searching. So basically microtubules can grow in a random direction, and if it doesn't find some target, it eventually shrinks rapidly, resets, and grows in another random direction until it finds the target. So it turns out that this switch from growth to shrinkage is not a simple Markovian process. It's a complicated multi-step process that might be optimized for this search process. And so what we have done is to come up with a discrete state stochastic model for the microtubule where the basic motifs are these non-equilibrium cycles of molecular reactions like GTP hydrolysis. And it turns out that this model hosts these topologically protected edge dynamics that reproduces this stochastic switching behavior and also this non-Markovian statistics of these dynamics. So if you're interested, please come by. Thank you.
That's everyone I had on my list. If there's more poster presenters, come on up. You'll have to say your name and your poster title.
All right. I'm Nicholas Clausen, and as you can see, I submitted my poster late. So the title of my poster is a mean field model for active plastic flow of epithelial tissue. So this is work on morphogenesis based on two ideas. Morphogenesis happens much slower than the speed of mechanical relaxation. So you can think of tissues as being an adiabatic force balance. And the dominant, most important source of stress in tissues is coming from active stresses regulated by biomechanical feedback loops instead of by constitutive relationships like in a spring. Based on these two assumptions, you can derive a very useful geometric representation of the network of active stresses in a tissue, about which you'll hear more in Fritjof's talk tomorrow at 11. And in my poster, I will use this perspective to derive a systematic mean field theory and identify the relevant variables that link together cell-level dynamics and tissue-scale outcomes. And we'll study a model of epithelial morphogenesis that shows how local mechanical feedback can couple to a cell-level geometry to encode tissue shape with applications to the fruit fly embryo, Drosophila melanogaster. Thanks.
Hi, my name is Satvik. I'm a graduate student in Zwanmir Dojik's lab, and I have a poster about an experimental system where we have a chaotic active fluid composed of microtubules and kinesin motors, and we mix in passive actin filaments and an actin crosslinker. And what we find is that the active fluid flows can drive the assembly of complex mechanical structure built out of the actin filaments. And once this structure is formed, the active fluid, which permeates this passive network, can drive elastic deformations that sometimes are very macroscopic, shear oscillations. So there's an interaction of active and passive materials across multiple length and time scales. If you want to see more about that, come see my poster. Thanks.
All right, fantastic. Let's give all of the lightning talk participants a round of applause. Amazing. Thanks to all the speakers for a wonderful first day. And